Greetings to all the students. Welcome to the free biology and chemistry revision classes. Our today's topic of discussion is enzymes, which is a very important topic of our pre-O levels, O levels class 7, 8, 9, 10, both at Excel and Cambridge. So now let's see. We will do a quick revision of this topic of enzymes. What are enzymes? They are actually the biological catalyst. You must have heard the name of catalyst. Okay. If you have not heard the name of catalyst, let me tell you what these catalysts are. What are the catalysts? So catalysts are actually, these are the chemicals which are used in chemistry when during a chemical reaction, sometimes the reaction slows down. So we put a chemical which is called a catalyst. It is a chemical compound. It can be a single element. It can be a chemical compound. It speeds up the chemical reactions, but it does not used up in the chemical reaction. It will not be finished in the chemical reaction. At the end of the reaction, you will get the catalyst. Exactly the same mass you have added, you will be able to retrieve the same mass at the end of the reaction. So what it has actually done, it has speed up the chemical reactions. So in our cells also, in the living organisms, chemical reactions are taking place day and night to maintain the life. What these chemical reactions are called, they are called metabolism. So there are two types of the metabolism, anabolism and catabolism. There are two types of the chemical reactions taking place in our cells day and night. What they are? Anabolism and catabolism. So in case of anabolism, you can say digestion is the digestion. Assimilation is the example of anabolism. What is assimilation? In assimilation, tiny molecules join together. They make a bond and they make a complex molecule. What happens in digestion when the glucose gets inside our cells? The glucose which is not used up, the tiny molecules of glucose join together and they make glycogen and store in our cells. So anabolism are that type of the chemical reactions in which the tiny molecules combine together and make a complex molecule. On the other hand, the another type of the metabolism is catabolism. What is catabolism? Digestion is catabolism. What happens in catabolism? The complex molecules are broken down into tiny, simplest molecules. For example, during the chemical reaction, starch is broken down. During the process of digestion, starch is broken down into glucose. This is catabolism. So no matter it is anabolism or it is catabolism, enzymes are needed to speed up the chemical reactions. And these chemical reactions, some of them, most of them are generating energy, some of them are consuming energy. So these chemical reactions are taking place in all the living cells of plants, animals, microscopic organisms, human beings. These chemical reactions are taking place, and the chemical reactions will be impossible if enzymes will not be there. They will become too slow if enzymes will not be there. All right. So what enzymes are actually made up of, from where they are coming in our cells? We are manufacturing enzymes in our cells, ribosomes. Ribosomes are the tiny organelles in our cells. We have got in plants and animals, we have got some tiny units having a special function. For example, nucleus is one of the organelle. Mitochondria is one of the organelle. Similarly, ribosomes are also tiny organelles which manufacture proteins. So from where they are getting the raw material for protein, whatever the proteins you are eating, in your digestive system, you are digesting the proteins into amino acids. Amino acids are released in the blood, and the blood will take these amino acids into your cells. Amino acids will be used up for your bodybuilding, muscle building, repair, healing. Some of the amino acids are used up by the ribosomes as a raw material to manufacture enzymes. So what actually enzymes are made up of? They are made up of proteins by the ribosomes. In our cells, we have got tiny units which are called ribosomes. So they are playing a role in manufacturing these enzymes. So these are some of the properties of enzymes, children. You can see they are very important. We should know the properties of enzymes, like they are made up of proteins. They speed up the chemical reaction. They are not used up in the chemical reaction. Just like a catalyst in chemistry, at the end of the reaction, you are going to retrieve it back. Similarly, you are going to get the catalyst at the end of the reaction. They are the way they are. They are sensitive to pH. In our cells, most of the cells, the pH is 7, the neutral pH. And at this pH, the enzymes are actively working. Increasing the pH or decreasing the pH will definitely cause the damage of enzymes. They are sensitive to temperature. Our body temperature, all the organisms, all the living creatures have a certain body temperature. Human beings and birds have a body temperature of 37 degrees Celsius, and this is the temperature at which the enzymes are most active. If the temperature goes up, the enzymes are destroyed. If the temperature goes down, they will slow down. They will become deactivated. And this is the reason that when we have fever, why do we feel so weak? Because our enzymes are denatured. Chemical reactions are not taking place faster. Energy is not generated. So when we recover in a very new way, once the enzymes are destroyed, we are not going to get them back. So when we recover, that time we are actually making new enzymes. The poisonous substances, if they make their way in our cells, they will destroy the enzymes then and there. Enzymes are specific in nature. It means one enzyme is meant for only one chemical reaction. One enzyme cannot be used in multiple reactions. Certain vitamins and minerals are needed for enzyme activity. Vitamins and minerals do not produce. We have done the chapter of diet, and we have seen that vitamins and minerals do not produce energy directly, but they are needed. Why? Because they boost up our enzymes. So certain vitamins and minerals are needed. This is the reason that when we are sick, when we are recovering, doctors prescribe us to have multivitamins, fruit juices. Why? To boost up the enzymes. Lock and key model of the enzyme and substrate activity. What does it mean? This means that every enzyme is going to act on a certain type of the substance, and that substance is called a substrate. For example, the enzyme amylase, which is starch digesting enzyme, it will act on starch. So amylase is the enzyme, and the substance on which it is acting upon, that is starch, is the substrate. All right. So amylase will act on substrate, starch. All right. How? What is the mechanism? You can see this shape of the enzyme. On the surface of the enzyme, there is a particular shape. And this particular area on the surface of the enzyme is called active site. An active site has got a particular shape. And this shape is exactly opposite, or you can say corresponding to the shape of the substrate. So you can see this is the enzyme in this picture, and this is the substrate molecule. This is just an example. So the substrate molecule can come and easily fit into the active site of the enzyme, just like a lock and key, just like a key fits into a lock. Why? Because the shape of the key is corresponding or opposite to the shape of the keyhole. That's why the key fits into the keyhole and opens the lock. Similarly, on the surface of the enzyme, there is a particular shape, there is a particular area which is called active site. An active site has got a particular shape which is corresponding to the shape of the substrate, the substance on which it is going to act. And the substrate fits into it just like a lock just like a key fits into a lock, or you can say just like the two pieces of jigsaw puzzle. And the next moment, what happens? It is going to break down the substance into tiny, simplest molecules. This is the lock and key model of enzyme and substrate. So you can understand, you can say that substrate is acting just like a key, and the active site of the enzyme is acting just like a lock. So they fit into each other and the chemical reaction happens. Why enzymes are specific in nature? Why one enzyme acts only on one kind of substance? Same reason. The reason is same. You can see one of the enzymes have got a surface area. One of the enzyme have got an area on its surface having this shape. Let me show it with another color pen so that you can understand. Suppose one enzyme has got an active site of this shape. Because the substrate on which it is going to act can easily fit into it, because this is also a square shape thing. This is just an example, remember. Another enzyme has got this type of the active site shape. So see, the same substrate cannot fit into it. So actually the shape of the active site decides what type of the substrate is going. Maybe the substrate which is going to fit into it should have a shape of something like this, should have a shape of something like this, and it will easily fit into it like this, and the reaction will happen. So why enzymes are specific in nature? Because every enzyme has got a different shape, which is corresponding to the shape of the substrate, and every substrate molecule has a different shape. So only the substrate which have a shape corresponding to the shape of the enzyme molecule can fit into it. This is the reason that one enzyme can act only on one kind of substance, because of the particular shape of the enzyme active site. Enzymes are sensitive to temperature. Why enzymes are sensitive to temperature? I told you that every organism has got a particular body temperature. For example, birds and human beings have got the body temperature somewhere plus minus around 37 degrees Celsius, and this temperature is called the optimum temperature. The temperature, maximum temperature at which the enzyme is most active. So at this temperature, the substrate will fit into the enzyme like this, and the reaction will happen. But if the temperature goes above 37 degrees Celsius, like if it reaches to 40 degrees Celsius, the shape of the active site changes. It is destroyed. So the substrate can no longer fit into it. The reaction will not happen. We say that the enzyme is denatured. The meaning of denatured means destroyed. The enzyme is destroyed. Why? Because increasing the temperature changes the shape of the active site, destroys the shape of the active site, and the substrate can no longer fit into it. So the enzyme is destroyed, and we are not going to get this enzyme back again. Then what we have to do? We have to manufacture it in our cells in a very new way. This is the reason that enzymes are sensitive to temperature. That if the body temperature goes below the optimum temperature, what happens then? I have explained you what happens when the body temperature goes above 37 degrees Celsius, above the optimum temperature. Similarly, if the body temperature drops down little bit from 37 to let's say 35 degrees Celsius, what will happen? The kinetic energy by which the enzyme and substrate will move and fit into each other, the kinetic energy will decrease, and they will move very slowly, and they will not get enough energy to fit into each other. They will try to fit into each other, they will try to bond into each other, but not getting enough energy to fix it over there. We say decreasing the temperature, the enzyme is not destroyed, the active site is not destroyed, but the enzyme has become deactivated. It means when the body temperature will again come back to the normal, again the enzyme will be active. Similarly, what happens when the pH changes? I told you that most of the cells have got a pH of 7. That is the optimum pH in most of the organisms. So if the pH changes, if it goes above or below, in either way, in either case, the enzyme's active site will be destroyed. All right, so you can see over here in this picture how changing the pH has changed the shape of the active site, and this substrate can no longer fit into it. So changing the pH or increasing or decreasing the pH will totally destroy or denature the enzymes. These are the examples of different enzymes like catalase, diastase, pectinase, pepsin, renin. You are familiar with this enzyme pepsin because we have done it in the digestion class, digestion episode which I have uploaded. So pepsin, salivary amylase, lipase, they are all the enzymes. Substrate on which they are acting, pepsin is acting on the substrate proteins, and and the product is the polypeptides or amino acids. So this is the short revision, children, of the chapter of enzyme. And in my next video, I will try to cover the experiments of enzymes. So that's all, children. I hope you will like this video and it will be helpful for you for your exam preparation. Bye bye. See you soon in the next class.